Kia ora koutou, ko Ange Fleming aho. Welcome everybody to this workshop today at Dignan Street Community Gardens, part of Growing Point, with funding from the WMIF grant from Auckland Council. We are going to talk today about diversity from kind of the beginning of composting right through to the end where the diversity's in the soil. So we're going to talk about all the stages of the food soil web as we go around from the compost. So we can start over here. If you'd like to come and gather around. This is the compost bin that we're working on at the moment. This is a hot composting bin, and we inoculate this bin with beneficial anaerobic microbes. It's very important that you put in a diverse range of inputs as well as microbes, because it's the microbes which are the primary decomposers. Everyone, when they think of compost, they often think of worms being the decomposers, but actually the worms eat the fungi and the bacteria that are decomposing the food in the beginning. But we use a lot of coffee chaff as our carbon source, and we use vegetable scraps and gardening clippings from a number of sources. So we've got a nice diverse range, and that is currently sitting at about 30 degrees. As the compost gets more mature and we get it to a bigger volume, once the bin gets full, it should get up to between 55 and 65 degrees for around about 2 or 3 days. And then we know that we've killed all the pathogens. If there's any pathogens present, they'll be killed off, and that creates a safe compost. It also doesn't get too hot, so you don't kill off all the good things. And then once that's matured, it gets turned, maybe just once, maybe twice. And cardboard as well. We mulch our own used pizza boxes, any kind of boxes contaminated with food that can't be processed in the normal council recycling way. So one of the measures that we look at for composting is the number of worms, and that's also true for looking at the number of worms in soil. So if we've got a really good healthy soil, we want about 30 worms per spade spit. Let's see what we can find. There's a good one, yeah.
Just feel around.
Yeah.
Oops, half one.
Where's the other half? 1, 2, 3, 4, 5, 6. That's it. It's actually pretty good. This bed's actually pretty good. We can see the soil's quite nicely flocculated as well, so that's and that's about the calcium and magnesium ratios. Okay, now we're going to do another spade spit worm count from the beds that have had a lot of input of biological microbes, such as beneficial anaerobic microbes, compost tea tinctures, what else? Protozoa teas. We've used the biologic steps 2, 3 and 4 on these as well. Expecting this to have more fungi and hopefully lots of worms. It's a bit of a small spade spit, but we'll have a look. soil's got a nice kind of crumbly structure to it. And oh, there's a potato. There's a worm. It's looking a bit nicely kind of aerated. I can see pockets in there. Lots of good roots. This one does have brassicas. This bed has a lot of brassicas in it, so we're unlikely to see a lot of mycorrhizal fungi because brassicas don't form that relationship. They've lost the genes for mycorrhizal fungi relationships. Still, there aren't heaps of worms, not as many as we saw on that other patch, which means these beds are still higher in bacteria than fungi, so we need to lift that. We still need to keep lifting that. There's a few there. What we're doing in the beds is polycropping in a really bio-intensive way. So for each bed, which is 1.2 metres wide, we have 6 rows in there, and we do a lot of planning about how to do that. And we're going to do a little exercise in a minute on doing that. So we start on the outside with things that we can cut, like this coriander here. And then as you go in, you've got to pluck, which is something you might pluck. This is purple sprouting broccoli, so you'd pluck the broccoli heads, and you'd go in another row and you'd have something that stays in the ground for a long time that you pull out, like fennel or radishes and that type of thing, and then you'd do the opposite on the way out. So the second exercise I wanted to have a go at is looking at planning and we've got a seed sowing guide for Auckland. If someone wants to hold that. And if we want to think and talk about what we'd like to grow in our garden next, the next thing to do is to have a look about when to sow things. And then we can plan. We try and keep our beds full, and that actually does involve a lot of planning and seed raising, and having plants ready to come along, thinking about when they're going to be harvested, and working back a month beforehand so that you've got plants to put in the garden when the other one comes out. And the more diverse the plants are on the top of the ground, they'll all be feeding the soil with their excess sugars that they make from their large solar panel leaves, and that they put some of that back into the soil and feed the microbes in the soil. I've got some pink ones for pulling and some yellow ones for plucking. And a pen for deciding what else you would like to grow in there. And these ones the blue ones are cut. We're doing cut, pluck, pull, gluck, cut. Can you see that, so that you can see that the middle of the beds get left alone because they're the hardest to reach. So that's where you put the pull things. And possibly they're the biggest. The next is pluck, which might be your tomatoes, basil, capsicums, anything you pluck. Zucchinis, corn, all that stuff is pluckable. And then cut. And this isn't hard and fast rules by any means. This is just trying to make it easier. So, but what we want is the diversity as well. So here we've got lettuce, guylan. We're not doing a whole row of one thing. And the thing about the brassicas is it's really important to mix your brassicas around because they don't form that relationship with the mycorrhizal fungi with their roots. So we really want to keep them separated. We kind of underplanted those brassicas with plants that like coriander, what else have we got in there? Some lettuces and things. That we did inoculate the seeds with mycorrhizal fungi so that it is getting into the soils. And so that's what we're thinking about going forward. The main thing about pest control is increasing the overall health of your plant. So if your plant is really healthy, it's getting a lot of calcium from the soil, it's getting a lot of silica. This is one more, I think it's boron. You'll have the silica is like, you know, like when plants are hairy, that's silica. And so those kind of plants are less attractive to insects as well because it's hard for them to get into the stem. And so calcium also plays a big part in cell wall structure. And so the better your cell wall, stronger your cell wall is, the harder it is for insects to penetrate that cell wall. And also the higher your sugar level, called the Brix level of your plants, also the insects don't like that. They don't have a pancreas, so if the sugar level's too high, it ferments in their stomachs and makes them sick and can even make them die. So by increasing the overall strength, the overall health of your soil, you'll have less weeds and you'll have more pest-resistant plants. So you can't lose in the strategy that we are going forward with.